The keywords that were significant in this analysis of the 2,531 that I mentioned are here. There are 21 of them, and I'll mention a couple of them. In particular, ethanol. In this case, the ethanol is one of the keywords in the vocabulary, so it serves as a positive control that we went looking for ethanol in this analysis, and in fact, we found it. That's the 263 citations that were in PubMed. We also queried PubMed for the other keywords that were found to be significant in the PDG ACE analysis and ranked them by the number of PubMed citations. The top three after ethanol are consumption, background, and intake. And upon review of the Entree gene records for these keywords, we find that consumption and intake both refer specifically to consumption and intake of alcohol, and background refers specifically to genetic background. So this combination of these top scoring keywords gives us an indication that this pair of genes is influencing or is being influenced by ethanol consumption or intake. So at this point, we're talking about not only a genetic interaction, but ethanol is an environmental influence on this genetic network. So we really have a combined gene by environment interaction. So we now have what we think are reasonable candidate genes for this genetic effect. We have evidence that these genes interact and that there is an environmental influence on this interaction as well. And the next step is to try to put this analysis into context. And we use GeneGo GeneGo software to perform this step. The database at GeneGo is called Metacore, and it has many protein-protein interactions, protein-DNA interactions, and protein small molecule interactions, as well as graphical software to provide a nice user interface and analysis tools that allow you to assess statistical significance. So I input just my two candidate genes, TNF and MTHFR. TNF is also called TNF alpha, and asked GeneGo to give me a network. It actually gave me several, but the highest scoring, the most significant network, was this very simple one with just 4 genes in it. And the way that this network works is TNF alpha, as I mentioned, is a signaling molecule. It binds TNF receptors and activates the receptors. The receptors activate NF-kappa B, which is a transcription factor, and NF-kappa B activates the transcription of TNF alpha. So you'll see the arrow goes around in a circle here, and this is in fact a genetic feedback loop. You can imagine that whatever influence that there might be on TNF alpha, it's transduced through TNF receptor to NF-kappa B, which then amplifies the signal to TNF alpha. And with each iteration through this cycle, the signal would grow stronger. Notably, as this signal is growing stronger, or for that matter weaker, it has an effect on MTHFR as well. I mentioned that MTHFR is a folate metabolizing enzyme, and so whatever happens within this network also influences MTHFR, which influences folate metabolism. So putting all of those pieces into context, and I should mention that I've turned the graphic. Here's TNF receptor, NF-kappa B, and MTHFR. When I added ethanol to this network, I find that ethanol exposure suppresses TNF alpha converting enzyme to reduce the amount of TNF alpha that is available. So as I mentioned, we have a feedback loop here. So with a reduction of the processing of TNF-alpha, we have a reduction in this signal, a reduction of this activation, a reduction of this activation, and a reduction of expression. So we now have two different influences on TNF-alpha to reduce this signaling cascade, which would be expected to reduce the expression of MTHFR, which is going to alter the availability of folate. And interestingly, low levels of folate are associated both with alcohol abuse and with depression. So at this point, we have a network that is consistent with this genetic influence by environmental interaction on folate metabolism, leading to increased susceptibility to depression. So when one of my co-authors saw this network, she immediately identified that network as a subnet of one carbon metabolism. Here's TNF, TNF receptor, NF-kappa B, and MTHFR. Well, there are actually additional enzymes that participate in folate metabolism, and you can see that every single one of them is also influenced by this same feedback loop, so that the effect of ethanol through this network actually would influence potentially all of these candidate genes, and genetic variation in any of these candidate genes could in fact influence genetic susceptibility to both alcohol use disorders and depression and the comorbidity. So we have a number of additional candidate genes based on that model. So I went back to PubMed and looked for evidence that any of them participate in this, or if there's already evidence that any of them participate in the comorbidity. And I did not find a lot of evidence. There are some, like BHMT, where we have some evidence of the influence on ethanol or an influence by ethanol, but there's not a lot of evidence already in the literature for these effects. In spite of that lack of evidence, we do have a model now that is consistent with long-standing dietary guidance of monitoring and potentially supplementation of folate for patients in treatment for depression, alcohol use disorders, or the comorbidity. And at this point, we have specific variants identified as risk variants. We could identify risk variants based on these candidate genes, and this is exactly what we need to do to identify candidates for the pharmacogenomic studies that are just now beginning to be implemented in psychiatry and in other disorders. And at that point, identifying the specific disease variants is likely to improve the prognosis for patients. So in conclusion, this approach really can be used for many complex diseases. I want to emphasize that in every step of the analysis, it's important to use the most reliable data. In this case, we did that to select candidate genes. Use multiple lines of evidence to test your hypotheses. And if at all possible, use independent lines of evidence. Then put the candidates into context. And at this point, we now have candidate genes where we could go back to genome-wide association or microarray data and use those data to test the hypotheses that specific variants are associated with the disease. And we end up with a model that puts the environmental effect of alcohol consumption into a genetic context and provides a model for further hypothesis testing. Thank you.